Hello ladies and gentlemen, welcome to our video on the introduction to digestion. Today we're going to be talking about the different types of macromolecules and eventually discuss how enzymes affected digestion of those macromolecules. Today your goal is to know all four of the macromolecules, identify the different structures such as dipeptide or a polysaccharide, and then identify what is the function of the macromolecules in the body. We will eventually get to chemical tests as we move through this chapter. We're going to start pretty basic with the introduction to biochemistry, so the difference between molecules and compounds. A molecule is formed when you bond two or more atoms together. A compound, on the other hand, is formed when you bond two or more molecules or different types of elements together. My example here is showing you a molecule can be something like oxygen atoms, O2, where a compound can be something more like water, H2O. Now this is important because as we move through this chapter, we're going to be discussing how we break down compounds. Sometimes we will talk about molecules, but mostly we're going to live in the world of compounds. Another quick introduction to this chapter is talking about the difference between inorganic and organic. Now, inorganic compounds don't contain carbon, which is really key because if we talk about something in your body and it has carbon and hydrogen, it's more or less going to be organic. Inorganic compounds do not contain carbon. Think of like NaCl, which is salts, water, H2O, or oxygen, okay? None of these have carbon in them, and especially not carbon bonded with hydrogen, so they are inorganic molecules. An organic molecule, on the other hand, it contains carbon and hydrogen bonded together, and they are going to be the things that we talk about in this chapter, macromolecules, carbs, fats, and proteins. So now to continue to build on this understanding, we're going to talk about how we build up or break down compounds. So we're going to start with this idea of dehydration synthesis. Dehydration synthesis is synthesis, so we know that means to build or make a larger molecule from smaller molecules. We accompany this by the loss of water, hence the words dehydration. So what's happening here is I have two examples. Two examples of glucose molecules. And on them, they have a hydroxide, the OH, and a hydrogen, the H. What's going to happen is those two are going to combine to become water, H2O. When they do, they create a reaction that causes those two molecules to come together to make a new compound called maltose. Maltose is a more complex sugar. So glucose is a very simple sugar. Maltose is a little bit more complex. Now, in the world of digestion, we're not going to see this very often. Dehydration synthesis is actually found in a process that we already know, photosynthesis, specifically the Calvin-Benson cycle. When we talked about making glucose, we talked about the Calvin-Benson cycle, taking 5 carbons, making 6 carbons, breaking down to 3, but then that 3 building back up to 5. Moving from 5 to 6 and 3 to 5 is the dehydration synthesis. Now, this is where we're going to spend most of our time talking about. We're going to talk about hydrolysis, the cutting or lysis of a larger molecule to create molecules by using water. So hydro, water, lysis, cutting. So we're going to use water to cut apart molecules. So let's go back to my example of maltose. So maltose is a compound made out of 2-glucose. It can be made smaller. So what ends up happening is water gets introduced right at the bond site there, and it eventually breaks it apart back into its 2-glucose molecules, adding back on the hydroxide and the hydrogen ion, okay? Now, it's important to go back, pause this, go back and draw these two pictures and make sure you have both these definitions in. You need to know dehydration synthesis and hydrolysis, both definition and drawing. With hydrolysis, this is the process that we're going to focus on in this chapter. This is what we would call digestion. We take larger pieces of food such as bread, which is a complex carbohydrate, and we break it down into the simple sugar such as glucose. And we know glucose is a big part of cellular respiration. So that's why we got to take it down to the really simple forms. So we now kind of finished the forefront of the ideas, the little basic stuff. Now we're going to really get into what are the macromolecules. The first one we're going to talk about is carbohydrates. Carbohydrates contain carbons, hydrogens, and oxygens always in the ratio of a 1 to 2 to 1. C6H12O6 or glucose. 6, 12, 6, 1 to 2 to 1. Carbohydrates provide us with short-term and long-term energy storage, which is really key. Now, if you look at the picture along the side, you're going to see something called a disaccharide and a monosaccharide. You need to know those words. They are key. Saccharide meaning sugar, mono meaning one, di meaning two. What you're going to see here is the breaking down process of a disaccharide to a monosaccharide, which is essentially digestion. All carbohydrates can be broken down into a simplest form to glucose, fructose, or galactose, the simplest sugars we have. They are all made up of a more complex sugar such as maltose, sucrose, or lactose. So what we have here is just an example of both the monosaccharides and the disaccharides. The other side to the story of carbohydrates are something called polysaccharides. Polysaccharides are more or less the carbohydrates we can think of. Breads, pastas, fruit even, okay? 3 very common types of polysaccharides are starches, glycogen, and cellulose. So starch and glycogen are the ones that we would actually use in the process of breaking down to smaller things like glucose. Starch is found in plants, so think potatoes, and glycogen is found in our muscles. Interestingly enough, cellulose is not something that we as humans can digest. Animals can in a long process, but cellulose is found in the cell wall and it's really tough to eat. We can't actually eat it. Hence why when you eat celery, you actually have you're burning calories eating celery rather than eating calories, because celery is made out of cellulose and we can't digest it. So we don't actually get any caloric value from it. Animals like cows, for instance, have extra stomachs because what they do is they chew on the grass to try to break down the cellulose. They swallow it. It gets turned up in their stomach. Then they kind of regurgitate it back up, and then they eat it again. And then they do that to pull out all the nutrients from it. So these are just examples of the types of things we're going to talk about. We're going to talk about polysaccharides, most complex, disaccharides, second complex, monosaccharides, the most simple. Now we're going to discuss fats, or what we would call lipids. This is another type of macromolecule, and they are insoluble in water. These actually give us way more energy than both proteins and carbs. So fats aren't bad. They get a bad rep, but they're not bad. Unsaturated fats are examples of like almonds, vegetables, fish, olive oil. They're good, okay? They raise good cholesterol in our body. Saturated and trans fats, not as much so. Saturated fats, though, in moderation are good for you. Like if you think of beef or butter, you can eat those as long as you don't eat a ton of them. Trans fats are more the ones that we think about when we think about like fats like cookies, donuts, cakes, fries, those kinds of things. So when it comes to fats, you need to know how they're formed. In their basic form, they're called 1 glycerol and 3 fatty acids. So you can see the green strip on the left-hand side there, that's a glycerol, and the 3 orange strips are all fatty acids. When they're combined together, as they are in the picture on the right, that is called a fat. In our body, we need to break those fats down back into their glycerol and fatty acids. We need to do a hydrolysis. So we're going to talk about how our body can break down into those 2 simple compounds, a glycerol and fatty acid. Now, a little bit more about saturated and unsaturated. You need to know that saturated just means that the carbons that are found on the chains are covered in hydrogen, whereas in unsaturated, there's less hydrogen. So if we look at the picture, you can see the saturated one is a long, straight portion with lots of hydrogen, where unsaturated is kind of kinked or bent. And that's because it's got a double bond between the carbons. Now, unsaturated is more liquid at room temperature, okay? And these are ones that are a little bit easier to break down. So proteins, one of the last few we're going to talk about. So proteins we're going to talk in depth about them. We're going to call them dipeptides, polypeptides, and amino acids, but we'll get in that next day. What I need you to know is that cellular structures of proteins are very different. You have what's called your structural proteins and then your globular proteins. Structural proteins are things we've seen already, such as muscle or connective tissue. Connective tissue being, think red blood cells, right? Muscles, we've seen branched muscles, tubular muscles, striated muscles. They all have structure to them. The globular proteins, on the other hand, don't. They're things like enzymes, hemoglobin, antibodies. They have a structure, but they're not like what we would think like a building, built like a building. They're more kind of random and abstract. This is an example of a hormone called insulin. And what you see in the gray picture are a bunch of amino acids, which I'll talk about in a second. And they're in this long chain, and then they're folded together and bound together. the picture on the right, which is colored, is insulin. Insulin is a globular protein. It's a hormone. And like I said, it's a little bit more random and abstract. It's not structured like you would think, like a muscle is. So the smallest form that you can get in proteins is called amino acids. They are the building blocks of our proteins. They build up proteins. As you build an amino acid, you make a specific protein. Proteins are found all throughout our body, so they're super important. Our body can make or synthesize 11 of the 20 amino acids that are out there. So there's only 20, so there's lots of different combinations. There are the other 9 that are essential that we don't make. So we have to make sure to eat a balanced diet because we need to make sure that we're getting those amino acids from our diet. This picture right here just shows a general structure of an amino acid. You don't need to know it, but it's just kind of interesting. Last but not least is the nucleic acids. Now, nucleic acids make up your DNA, which makes up your chromosomes, which makes up you. You're made out of sets of 23 chromosomes, okay? So that's really important to know. Okay, and we have pairs, and they're double-stranded, and they're bound together with A's, T's, C's, and G's. There's so many cool things about DNA. We're going to get that whole thing going in Bio 30. For now, you just need to know that nucleic acids can be broken down, okay, and that they make up part of your DNA. But we will not be talking about them through the process of digestion, but you do need to know that they are part of the 4 macromolecules. So make sure you have everything in your notes, and if you have any questions, please let me know.